All right, can you guys hear me? Is the mic working? Yes? No? 2 people nodded, so I'll take that as a yes. Welcome to SI. First, I'd like to point out that after 15 minutes of staring at the same meme, I think they look like a really cheap version of the Fast and Furious cast, you know what I'm saying? Like he's, what's his face, Paul Walker, and she's, I don't remember her name, his wife. I think they look like them. Anyway, I hope you enjoyed the jokes. You can leave now. That's all I had planned. The slides just got posted to Canvas as an announcement, so if you want to follow along, want to look at them later, want to pay me money for how much hard work I put into these 2 years ago, you may do so. For those of you who have not been to one of my sessions before, I'm Parker. I am a space enthusiast and resident Lord of the Rings expert, so now you know I'm a nerd. I love space. Ironically, my major is human biology and has nothing to do with space, but several of my electives have been in space-related courses. Interstellar physics was one of my favorites. I just like space. I just think it's a good time. I also love Lord of the Rings. Interesting fact for you guys. Since I was about 14 years old, I told my mom that when I got married, that I was going to insist that my wedding band be the one true ring from the Lord of the Rings. Everybody laughs. Everybody, thinks it's hilarious. The only person on this planet who's a bigger Lord of the Rings fan than me is my wife, and she actually just ordered me that ring, so you can suck it. In case you were wondering. We're just perfect for each other. That's all I'm trying to tell you guys. All right, let's get started with our interesting biology fact of the day. Everyone go ahead and try it. I already know you're going to. Your thumb is the same length as your nose. Put it right there between your eyes, and the end of your nose will be right at the end of your thumb. Your body has all kinds of crazy proportions like this. Your arm to leg ratio, the distance around your skull is the distance around your shoulders, all kinds of crazy stuff like that. We're actually more well proportioned than I may look standing up here in front of you guys, right? Yeah. So I wanted to get us started off with this clicker question because this was posed in class either this morning or Friday morning. I don't remember. I lose track. And not a ton of people got it right. It's only like 50% of the class got it right. And I felt bad because I talked about this in my session. And then I had like 115 people in that session. And then not very many people got it right. So I was like, maybe we'll talk about this again. So take a second. Which of the following statements about hydrogen bonds is false? Talk with your neighbor. Look at your notes. Do whatever you got to do. Try and figure out which one's the right answer. If you were paying attention in class, you should already know it. And then we'll go through it if you guys aren't feeling confident in it. This is just me fixing my conscience and feeling bad about not having explained this well enough. Okay, so who wants to say it's A? Or B? C? D? E? Who gave up after everybody said B and you were like, oh, it's got to be B then, even though I didn't know it. Everybody else seemed to know it. Okay. These format over so weird. I made this at midnight while I was drunk. No. It does this when I put it up there, so it's not my fault. Who can explain to me why it is B? Who got that right and wants to tell me why?
When 2 hydrogen atoms bond together, it's a covalent bond. Hydrogen bonds is usually between like electronegative elements, so like oxygen, fluorine, nitrogen.
Yes. So a bond between 2 hydrogen atoms would actually just be a covalent bond, right? Come on. A hydrogen to hydrogen. That's just a covalent bond right there. They're just sharing electrons. So let's see. A is true, right? Hydrogen bonds are weaker than covalent bonds. Hydrogen bonds are pretty much one of the weakest bonds out there. Hydrogen bonds form between 2 water molecules. That's true. Hydrogen bonds form because of polar covalent bonds, right? Hydrogen bonds are just a type of polar covalent bond. He mentioned, I think I heard you say nitrogen, oxygen, and fluorine. So I'll show you in this next slide here. It's between a hydrogen and either a nitrogen, an oxygen, or a fluorine, right? This is the hydrogen bond, okay? That's what that's saying. And hydrogen bonds give water many of its unique properties. That's the whole cohesive property with the water skeeter on the surface and the high latent heat of vaporization and how the swamp cooler works, and all those are because of hydrogen bonds. Okay, you guys feeling pretty comfortable with this? More than half of you answered correctly right here. This is going to come up on the test, I just about guarantee it. I haven't had a chance to look at his sample test yet. I'm going to so that I can talk about it in my super session on Wednesday, but I guarantee something like this is probably going to be on there. So watch out for that. All right, let's start on actual class material, and then I have an activity for like the last 10 minutes of the session. So if you guys notice me getting out of control, just throw whatever's heaviest, and whoever hits me neck above wins. Do you have a question? No? You're just pretending to throw. Somebody has to. I'll lose track of time. All right. 4 classes of organic molecules: carbohydrates, lipids, proteins, nucleic acids. Those are the 4 organic molecules that you can group everything into, right? Proteins, like a nice old steak here. Carbs being, what you usually think of, bread. Fats are in, a lot of dairy products, a lot of everything we eat in the United States. That's just who we are. Okay, so the first thing we have to talk about before we even get into these individual classes of organic molecules are 2 vocabulary words: polymers and monomers. Poly means many, mono means single. Okay, so a polymer is a large molecule of a bunch of repeated single units, right? A monomer is one of those units, right? I always compare it to like a pearl necklace, right? Each of the pearls is a monomer. All of the pearls together are a pearl necklace, right? So if you had 1 pearl, could you say you have a pearl necklace? So if you have 1 amino acid, can you say you have a protein? No. But you link up a bunch of amino acids, you link up a bunch of fatty acids, and you get, well, I guess fatty acids, but you get what I'm saying. You link up a bunch of amino acids, a bunch of carbs, whatever, and you get your full unit, right? This is, for whatever reason, this is a tricky vocabulary word for everyone. I always see students having a hard time with it, like on the test and whatnot. Just put it into whatever your own terms you have to have it in, right? There's lots of seats, guys, if you still want to come in. If you want to sit on the slanted floor like a weirdo, do it. So just compare it to a necklace, compare it to one of those candy necklaces, those things that they're like Smarties on it, but they're not Smarties. Do you guys know what I'm talking about? Yeah. Okay, those things are dope. I don't know, necklaces is all I've got apparently, but make up something, some kind of, like a chain on a bike, and each of the little bits is, I don't know, I saw the word chain. Come up with your own example so that you don't get them confused, okay? Because if you get them backwards, you're gonna get every single test question wrong, right? Is anyone confused on what these are? All right, I'm just gonna take that as a no. So how do we form them, right? You can't just, you don't just take an amino acid and like a puzzle piece, just stick it on another amino acid, right? You don't just take a pearl and stick it next to another pearl and call it a necklace, okay? Something has to link them together. This is a dehydration reaction. Why is it called a dehydration reaction? Taking water out, right? So one of the monomers has a hydroxide on the end, another monomer has a hydrogen on the end, and you stick them together and you get H2O, water, right? Do you see that? 1H, 2HO, yeah? So the actual polymer is getting dehydrated. So you think like dehydration means lack of water, So, but we just got water. How is that dehydration? You just have to think of it from the perspective of the polymer. The polymer got dehydrated, and the water left, right? And that's how you stick each monomer to the next one. Okay? Lots of blank stares. You guys are the best. All right, so let's talk about polysaccharides. What class do these fall into out of the four? Carbohydrates. Okay. So there are two purposes to carbohydrates and four kinds of polysaccharides, right? That's the polymer of carbohydrates. So we get energy storage and structural support. What does energy storage mean? It's how we store our energy, right? You eat bread and then you digest it and you get sugars out of it, get carbs out of it, and you store that in your energy and then you can use it later, okay? Structural support, what does that mean? Structure, as in cell walls of plants, right? So trees are relatively solid objects, right? You can't just walk over and like push over a full-grown oak tree, right? They're not bendy because they are built up out of one of these four. So let's look at them. Does anybody remember? Oh, I guess it kind of gives it away because the letters are there. Is starch for energy storage or structural support? Energy storage. And they come from plants or animals? Plants. I don't know why the letters are there. Pretend like they're not. All right. Yeah, so starch. Where do we get starch from? Throw out some foods. Potatoes, corn, rice, bread, all that good stuff, right? Cellulose then, structural support, right? Also some from plants. Anybody think of a food that's really rich in cellulose? Celery, right? Anybody ever heard that celery is like a toothbrush for your intestines? Anybody ever made the mistake of eating like a lot of celery at once? I haven't. Yeah, so that's what gives celery its rigid structure, right? You snap a stick of celery, you get a good hard crunch out of it. It's built up of a lot of cellulose. We can't digest cellulose, right? It's for structure, not energy. That's why it just slides right through. You get 0 calories from celery, and you spend, I think it's about 5 calories to digest celery. So if you only ate celery, one of the many reasons you would die within a couple of days would be there's nothing in it for you. You can't digest that. Also, like vitamins and minerals and stuff, and also it's just yeah. Questions so far? Glycogen, which energy storage or structural support? Energy storage from animals or plants? Animals, right? So when you this is how we as animals store our energy, right? So you might be thinking, well, I go and I eat steak and there's no starch in a steak because it's an animal, so how do I get energy out of it? Glycogen, right? You digest the energy storage that animal had and now it's yours, right? And chitin, structural support from animals. Can anybody name an animal with chitin? Somebody said crabs. Somebody else said butterflies, right? Well, lots of insects. Fungi, which are not technically a plant or animal. They're their own thing, also made of chitin. So technically mushrooms are more closely related to butterflies than they are to trees, in case you were wondering. Crabs, all those kinds of things, right? Anybody want to remind me the fun fact that Dr. Mott shared with us about chitin? I think it's pretty cool.
It's used for surgical growth.
Yeah. So our body can digest chitin, but like pretty slowly, and we're not like getting any nutrients out of it, just the acidity in your blood dissolves it, right? So if you have to get subdermal tissues, it would kind of defeat the purpose to stitch you closed and then stitch you closed on top, but then have to like cut you back open to undo the stitches. Plus, it's not like they can wait and do like one layer at a time, right? So they do the subdermal tissues with chitin. It dissolves really slowly on its own, and nobody has to go in and cut it out, right? So it's pretty dope. It's basically glorified butterfly poop just twirled around in your muscles. So pretty sweet too. Nah, it's not really butterfly poop. It'd be like butterfly legs or something. It's just less funny to say that, and nobody laughed anyway, so I don't care. Yeah, so we already talked about this. Cellulose makes up the plant walls. Most eukaryotic, pretty much all eukaryotic organisms that we know of cannot digest it, but some eukaryotic organisms have prokaryotes inside them that digest it for them. So cows, right? They eat grass all day. Cows actually can't digest grass, but prokaryotes in their stomachs can digest grass, and then they get the energy out of it, right? They just steal it. So that's how that works. Chitin, already talked about it. It's how they make surgical tissues, makes up the exoskeletons of a lot of insects. Okay, questions on carbohydrates before we move on?
Do you need to know anything about like amylose or amylopectin?
No.
Okay.
There's still quite a few seats if anybody wants to. You're just making a nice little line there. All right. Do what you want. Other questions? No? Yes? No? He's looking for you. All right, well, let's talk about lipids. What are lipids? Fats is one of them. They're also oils and phospholipids and steroids. You guys, this is like pulling teeth. Good night. We don't worry about steroids in this class. We just want you to know that steroids are a fat, and that's it. Like nothing else comes up. Don't study them at all, okay? Okay, so we'll get into fats and oils and phospholipids. I have two fats here, and I need you to tell me which one is saturated, which one is unsaturated, okay? So who thinks the top one is saturated? Who thinks it's unsaturated? Who didn't say anything because everybody answered saturated? You did. Now, did you have a question?
What's the difference between them?
We'll look at it right once I torture you with my questions. We'll actually discuss it. Okay. So yes, the top one is a saturated fatty acid. So by logical reasoning, the bottom one is unsaturated. Who wants to tell me? Who can kind of answer her question here? What's one or the other?
Saturated is saturated with hydrogen, and
Yes.
unsaturated is not.
So see how each carbon has a hydrogen? Well, except for this one. We don't care about him. And then on an unsaturated, you only have them here. You have an empty space here. Because these carbons double bonded to each other, it forms a bend in the molecule. I guess it's a compound. And that's what makes it unsaturated. It's called unsaturated because it's missing the hydrogens. The difference being its actual physical shape is what matters. So that bend is what gives it all the different properties. And we'll get into those in a second, right? Does that answer your question pretty much? Okay. Other questions before we actually talk about their differences? Before I forget, I'm going to pass around the roll. If you go to class at 8:30, put your name on the 8:30 side. If you go to class at 9:30, put the 9:30 side. Is there still like an overflow in the geology? Is that okay? So if you were in that one, do you go to 8:30 or 9:30 now? Either one. I don't care then. Put down your name on the hour that you go to class and your A number, and you'll be entered to win a prize. That's also not true. There's no free food and there's no lottery. Welcome to college. It sucks. No, College is great. I work for the university. I'm probably not allowed to say that into a microphone. Okay, so let's talk about saturated versus unsaturated. Okay, so saturated doesn't have any of the double bonded carbons, right? So every carbon has a hydrogen. They tend to be solid at room temperature, so these are things like lard and butter, right? So they tend to come from animals. I don't know why I'm pointing. They finally updated this computer and I can draw on it. Oh, jeez. Apparently not. So the saturated fats will usually come from animals, right? The exception being, what's it called? I'm blanking on. Margarine is technically from plants, but there's goodness knows what in there, and it's solid at room temperature. You should be worried. No. It's just they mess with it. What don't we mess with? Unsaturated fats, on the other hand, have one or more double bonded carbon. Okay, that's what this is. That's the double bonded carbon it's referring to. They'll be liquid at room temperature, and they come from plants, right? Corn oil, vegetable oil, peanut oil, literally any oil that has a vegetable name in front of it. I could go on and on, right? I don't think olives, what are olives? Are they like a legume? Somebody Google it, I dare you. Are olives fruit or vegetable or nut or what? I heard two answers. It's going to go down. Somebody Google it. I don't trust any of you.
You trust Google.
I trust Google way more. Anyway, questions about this. Olive oil is liquid at room temperature if you're not aware. No questions? Everyone's really feeling it? Huh?
Google says it's a fruit.
Google says it's a fruit. But I don't know why we're doing it. It comes from a plant. It's not vegetable fats. It's plant fats. So yeah, but thank you. Anything that says oil and has a plant in front of it, then okay. I'm an idiot. Questions? You all already knew that. All right, so this is what a phospholipid would look like. These are your fatty acids down here. This is your glycerol, right? Anybody heard of a triglyceride? Sound familiar? That would be if there was a third fatty acid right there, right? That would be a triglyceride, except that's not what this is, but you see what I'm saying. Then you have a phosphate. That's what makes it a phospholipid. And then you'll have some other group, usually some kind of charge to it. So this is the physical makeup of a phospholipid. Now, which end is, let's define some words. They're not up there. Hydrophobic, what does that mean? Water-fearing, right? Hydrophilic, water-loving. Which end is hydrophobic? Fatty acids are. Which ends are hydrophilic then? The phosphate group and the other group up there. So if you make a phospholipid bilayer, which is what most of our cells have as kind of their external membrane, right? Which end's going to be facing which? So the heads will be facing out of the cell or into the cell? Out. Oh, this is a terrible question. It's both. I did not think this through. Because a phospholipid bilayer has all the heads like this and like this. So I'm just going to answer the question for you. Those lines are the fatty acids. So the fatty acids point in, and the phospholipid heads are kind of on the outside protecting it, right? Because there's water out here and water in here. So the only safe place for these hydrophobic fatty acids is between the heads, right? That makes sense? I'm getting like ahead of myself and behind myself at the same time. I don't know. You guys understand what I'm saying. You're smart children. Okay. That's it for lipids. If you have any questions on them, speak now.
What does the bend in the unsaturated fatty acid do?
It changes the shape of the molecule. so it changes some of its properties. It melts faster, so that's why it's, liquid at room temperature. That's the only one I can think of off the top of my head. But yeah, it just changes the shape, and that changes its actual physical properties. So it's it's liquid at room temperature. That's for this class good enough. Got it?
So a phospholipid, is that just one that is water loving and water bearing and combined with fat?
Yeah. So in order to be this is just a saturated fat, right? But if you combine one of those with a phosphate and that charged group, it's the phosphate that takes it from just being a lipid and makes it a phospholipid. Does that make sense? It's got to have that phosphate on it. Back there?
Glycerol also non-polar in addition to the fatty acids?
Glycerol non-polar? I don't know, because carbon is just uncharged. So I don't think it counts either way, but I'm not sure. I just know that the fatty acids are positively charged, they're hydrophobic. The phosphates are negatively charged, hydrophilic.
Does it matter if the fatty acids are unsaturated or saturated?
For a phospholipid? Not that I know of, no. That just matters more in its basic state for what kind of fat it'll be. Other questions? Proteins. There's a lot more that goes into protein than most of you gym rats probably think. It's not just a powder that you buy from Costco and drink to make yourself think you're stronger. You're not. I just hate protein shakes, so I make fun of them in here. If you drink protein shakes, you're fine. I think they taste nasty. And I've tried every kind. And so someone's going to come up to me after and be like, bro, you got to try my protein shake. Tried every single kind on earth. Don't even try it. So the monomer is what? The individual unit, the monomer is amino acid. Put a bunch of amino acids together, you get a polypeptide chain, so that's the polymer. This one's fairly easy because they have the same prefix. And then you get a bunch of polypeptide chains together. You have a protein, right? So they work as catalysts. They speed up reactions, right? Enzymes are proteins. They are there for structural support, i.e. your muscles. They're involved in signaling, so like a lot of the neurotransmitters that you'll talk about in like a lot of your psychology classes and stuff. How many of you recognize like serotonin and stuff like that? Those are proteins. Defense, so red and white blood cells are proteins. Movement, again, muscles. And storage, a small amount of energy can be stored in your muscles. Most of it's stored in fat, but some is in muscle. Questions there? I know this is an extremely wordy slide, so don't try and write it down. Like I said, I posted this online. If this doesn't help you, completely just ignore it, but this is how I was able to memorize the 4 levels of protein structure, right? I have to compare stuff to real world stuff for it to kind of make sense for me. So primary structure is the order of amino acids that's going to determine what kind of protein it is. I compare that to the alphabet, right, that you learn in preschool. Your amino acids are your A's, B's, C's, D's, E's, F's, G's, right? It's just, it's an individual letter, okay? That's the amino acid, the order of amino acids. The secondary structure is the kind of folding that a chain of these amino acids does. It can either fold into what's called an alpha helix or into a beta pleated sheet, okay? I consider this like learning to organize the alphabet into words, right? So you take your amino acids, you take a C, an A, and a T, and now you have cat, right? Individually, that's just a C, that's just an A, and that's just a T. But you actually put them in a specific order, and you get a specific word, right? So depending on how you fold your amino acids, you can get one of your 2 structures, okay? Your secondary structures. Tertiary structure then is how all of the alpha helixes and beta pleated sheets in a whole group fold around each other. Okay, this is like sentences. Okay, so then you take cat. My cat is fat, right? You've strung together multiple secondary structures and got a tertiary structure. You string together a bunch of words, you get a full sentence, right? Quaternary structure, not every protein will have quaternary structure. It's just if that specific protein needs more, then you'll build on top of that. So it's like paragraphs. So you take my cat is fat, my cat eats a lot of food, my cat doesn't like people, and you can build a whole paragraph, right? Now, if you're talking about cats, you probably don't need a whole paragraph because I don't care. That's mean. So you don't have to have quaternary structure unless that's, the protein needs to go to that level. Most of them can just hit a tertiary structure, call it good. But they do have to have the first three. Okay, otherwise it's not a fully formed protein. Did I confuse you more, or was this somewhat helpful? 2 people said helpful. I'm going to consider it helpful.
Wait, so they have to have all 3 in order to be protein?
Yes. Yeah. They until they've reached that third structure, they're just not a fully formed protein yet. But they will eventually.
So they're in like the primary, the secondary, they're just polypeptide chains?
Yeah, and they're still on their way to getting fully formed.
So is the only difference between the tertiary and the quaternary just like the size?
Basically, it's like, how do I explain that? It's like the tertiary structure and another tertiary structure and how they interact with each other. So not every tertiary structure is going to interact with other ones, but if they do, that's how you get a quaternary structure. Other questions? This is kind of, oh yeah.
The tertiary structure is that's like the leftover. Like I would say it's leftover, but it kind of looks random, right?
Yeah, so you get like all your alpha helixes and all your beta pleated sheets and you fold them all over the place, and that whole hodgepodge is the tertiary structure. This is about as deep of knowledge as you need to go into this. Don't try and learn every single, just know the 4 levels, what makes up that level. So primary structure is made up of amino acids. Secondary structure is made up of alpha helixes and beta pleated sheets. Tertiary structure is, how those alpha helixes and beta pleated sheets are all wrapped around each other. And then quaternary structure would be how the tertiary structures are folded around each other even further, right? That's about as much as you need to know. Okay? Questions? So this is kind of a drawing of what they would look like. Your amino acids are just each of these little beads on the string, right? Those are your monomers. And then you take this whole big long chain and you fold it into a helix or into a pleated sheet. That's how you get that. Then you fold your beta pleated sheets and your alpha helixes around each other and connect them to each other, and you get a whole tertiary structure. Then you take another tertiary structure on top of that one, and you mix those together, right? So it's just ascending levels.
When you say fold, what does that mean?
Literally, they actually bend into shapes. So an alpha helix actually is helical, and a beta pleated sheet really is like a back and forth, crinkle cut fry. That's what those are called. You guys see what I'm saying? Don't act like you don't know.
So when we talk about muscles and the myofilaments in there, how do the myofilaments fit into tertiary and quaternary structures?
Myofilaments, you're talking about actin and myosin and all that. Shoot, you'd think I'd remember. We're going to get to that. I don't remember at what point we talk about that, but I know we do. I don't know. But I'll know when he talks about it in class. And then you'll know, and I'm useless. But we haven't talked about those yet, so not something to concern yourself with yet. If he talks about it in class and you're still wait, I don't know where those fit in, then we'll talk about it, if I ever figure it out. Other questions? So prions or prions, however you want to call them, these are those proteinaceous infectious particles that we talked about in class today, right? Bovine spongiform encephalopathy is one of them. You may now all applaud me for being able to pronounce that on my first try. Thank you. Yes, I know. I meant it, gosh dang it. That's enough. Scrapie is the sheep equivalent. Chronic wasting disease is the elk and deer equivalent. They found one in humans called Creutzfeldt-Jakob. Again, just it dissolves the proteins in your brain. Okay, so mad cow disease. They called it mad cow disease because the cows really got mad. Like they ran around all over the place and they tried to kill people, right? And these sheep rub up against barbed wire fences and don't even care that it rips their skin and their wool off, right? Because their brains are literally falling apart. Chronic wasting disease, same idea. Elk and deer will like break their own horns off on trees and just like rip their own heads apart because their brains stop working. Creutzfeldt-Jakob, it's a psychological disorder where your brain is falling apart, kind of like Alzheimer's. Alzheimer's isn't a prion. They don't know what Alzheimer's is, but Creutzfeldt-Jakob has similar symptoms, and it's just these are misformed proteins, right? They've bent in the wrong shape, right? But they're infectious proteins, meaning they work almost like zombies. If a prion, which is just a misshapen protein, touches a normal protein, it like infects that protein, and the normal healthy protein misfolds too, and it turns into a prion. And then that one touches another one, and it's like The Walking Dead, right? Just on top of each other. You guys know what I'm talking about.
So do they occur by mutation or?
I don't know how they start. No one knows. No, I'm just kidding. I don't know. Somebody might not. I just assume if I don't know, nobody does, right? No, I don't know how they start. I just know that they're misshapen proteins. They touch another protein, and they do that cascading effect where they infect more and more. Someone in the back had a question, I think, and then we'll come to you. Did I answer it? Did you give up? All right.
So it's nothing to do with like DNA or anything like that?
It might be that you have a mutation in your DNA, so you make bad proteins. It might be
But like from
you get struck by lightning.
good protein, the bad doesn't infect good with bad DNA.
No, it's protein. Its amino acids interact with the other protein's amino acids and kind of confuse them, and they fold wrong, right? Other questions?
What are the similarities between the products in humans and like CT and stuff?
I don't know enough about those to give you a confident answer because people always Google afterwards and they tell me I'm wrong, and I get called out and people stop showing up now. I don't know. If you felt like it, Creutzfeldt-Jakob's actually kind of an interesting disease. You can go home and research it, and then you can compare it because, like I said, it's kind of like Alzheimer's. It may have similar to CT and things like that. So it's always something to check out. You won't be tested on anything like that, though.
Are we going to go over the cell biology?
The what?
The cell biology?
Yes, that's in 2 minutes, and nobody had to throw anything at me. Other questions? We got nucleic acids, and then that's, I believe, when we start on cell biology. Yes, okay. So nucleic acids, they are the polymer of nucleotides, right? So a bunch of nucleotides makes a nucleic acid. These make up your DNA and RNA. That's where the name deoxyribonucleic acid comes from, is that they're made of nucleic acids. They don't have oxygen. They've had an oxygen removed, better said. Ribonucleic acids haven't had an oxygen removed, but again, they're made up of nucleic acids. These contain your genetic information. If you didn't know that about DNA, you probably shouldn't be in this class. No, you should be in this class. This is where we're going to teach you about DNA. They're held together in the classic double helix, like in that picture, and like in the thing when Jimmy Neutron gets like an idea and you see his DNA. Isn't that, he knows what I'm talking about. That's real, right? They actually form double helixes, and there's a hydrogen bond between the two outer strands, right? Which is actually really important, and we'll get into it more when we talk about genetics. But because hydrogen bonds are pretty weak, that makes them easy to break. Why would you want your DNA to be easy to break? So that you can open it up and copy more DNA, right? But we'll get into that. I just wanted to show you guys hydrogen bonds are super important, right? We already talked about the 5 essential properties, well, the 5 special properties of water. There's a 6th property from hydrogen bonds that's super important. Your DNA is held together by hydrogen bonds. You would literally fall apart without hydrogen bonds, okay? So they're pretty important. Questions about nucleic acids? And because I am such a generous and thoughtful and caring supplemental instruction leader, I made you guys a cheat sheet. Again, copy it now, take a picture of it now. I posted them online, so if you don't want to do that now, you can always just download the slides later, whatever you want. This is just my quick cheat sheet so that you can look real quick and just compare them out, right? So the molecule, carbs, lipids, proteins, nucleic acids, they're monomers and they're polymers. Important to note, lipids do not form a polymer. They form just their monomers of glycerol and fatty acids, right? A phospholipid bilayer is not a polymer, okay? Because they're not actually linked together. They're just next to each other. They just neighbor up because of their polarity, right? But they're not forming a bond. There wasn't a dehydration reaction, so it's not a polymer, okay? I totally messed that up last year, and I thought they were going to crucify me. It was nuts. I got like 5 emails, and people were like, what? And I'm like, I don't know. I wasn't paying attention in class. Neither were you. No. I got confused. They do not form polymers. Questions up to here before we start on cell biology? All right, so this is where we're going to do a fun little activity. Oh, well, we'll start the activity 2 slides from now. So let's talk about the 2 kinds. We got prokaryotic and eukaryotic. Which one's smaller? Prokaryotic. Which one's more abundant? Prokaryotic, right? There are far more prokaryotes in this world than there are eukaryotes, and someday they're going to take over. Eukaryotes, larger, less abundant. We are eukaryotic, right? So prokaryotes don't have a nucleus, right? They just have like a nuclear region where their DNA is just kind of, but it's not contained in a nucleus. They have very little internal membrane, right? They just have like a couple of layers here, and that's about it. And they have very few organelles, right? They have some ribosomes, nucleoids. That's technically not even an organelle. That's just a region. Right? They don't have a whole lot going on. They're very simple, very small cells. Eukaryotic cells all have a nucleus except for red blood cells. Now in this class, whenever you see an exception to the rule, pay attention. Dr. Mott loves those, right? He loves to go, all eukaryotic cells have nuclei, true or false? False. Red blood cells are an exception. They have extensive internal membranes, right? So they got insane membranes all over, right? And they have numerous complex organelles, okay? They got lysosomes, mitochondria, Golgi apparatus, ribosomes, microvilli, centrosomes, all of it, right? They're far more complicated. They're far larger, and they perform a lot more processes. So they need a lot more going on in the middle. Questions? All right, so unique to animal cells, animal eukaryotic cells, they have lysosomes, centrioles, and flagella. Plant cells do not. Okay? Plant cells have chloroplast, central vacuole, cell wall, and plasmodesmata. Animal cells do not, right? So these are exclusive of each other. So animals and plants both have, nucleus and rough ER and smooth ER and ribosomes and Golgi apparatus. They all have all that in common. Mitochondria, all that, right? But only animal cells have these 3, and only plant cells have those 4. I guess we'll. I'm trying to think if there's any big. No, we're just going to keep going. Questions? Because we're going to talk about some of these.
So that you talked about in class that animal and plant cells both have flagella.
He did say that in class?
Yeah. So eukaryotic. Okay, you're okay.
No, I'm right.
What's the flagella?
It's just flagella is the plural.
Oh, okay.
Yeah. So like mitochondrion is a single one, and mitochondria are multiple. It's like Latin. Doesn't make any sense. So yes, animal cells have flagella. Plant cells do not. What you probably were thinking of is that eukaryotes have flagella, while prokaryotes also have flagella. But not all eukaryotic cells have flagella. Other questions? I saw another hand, I thought. No? No? Yeah.
Do like all the organelles, like are they specific to each type of cell? So like we have multiple types of cells in our bodies. Are they specific organelles per cell?
Yeah, like like your red blood cells don't have nuclei. So yeah, this is like a generic animal cell. It'll differ by the animal. It'll differ by what kind of tissue it's found in, muscle versus nerve, all that, yeah. This is just the generic, what they have. Other questions? Okay, this is where we're going to do our activity. We may not have as much time as I would have hoped, but I have 5 cellular structures here. Okay, so we're going to kind of group up. How many rows do we have? 10. So the first 2 rows can be a group, and then rows 3 and 4 are a group, rows 5 and 6 are a group, rows 7 and 8 are a group, rows 9 and 10 are a group, and hall or passageway sitters can just go with 9 and 10. Okay, so group 1, I want you guys to decide what this is, okay? And then group 2, you get to decide what this is. Group 3, you get to decide what this is. Group 4 decides what this is. And group 5 decides what this is, okay? Again, these slides are on Canvas, so if you didn't get a great look at yours, which you probably didn't because we're out of time, talk amongst yourselves, decide what they are, what they do, and then make up sort of a test-type question that you think you could ask about whatever thing you have, okay? We'll go for a couple minutes, and then we'll ask for like a spokesperson from your group or something. Take your time. All right. We're running out of time, and people are starting to bail because they consider 5:50 a hard deadline. The person in here after me probably also considers that a hard deadline. So group 1, what are we looking at here? It's a ribosome. Okay, what do? They make proteins, right? So they string all those amino acids together, and they form a polypeptide chain, right? They can either be stuck in your ER or just floating around in the cell, either way. Ribosomes. Okay, group 2. What are we dealing with here?
ER.
your endoplasmic reticulum. Two kinds, smooth ER, rough ER. The smooth ER is literally called the smooth ER because it has no texture to it, right? The rough ER is considered rough because of all the little ribosomes stuck in it, right? It's like the backside of a crunch bar. It's just got all the little ribosomes or like little bumps in it, right? You know what I'm talking about. The smooth ER is for membrane synthesis. The rough ER is for protein synthesis, kind of like a ribosome because they have ribosomes stuck in them, right? Group 3, what are we looking at here? The nucleus, okay? So your DNA and your proteins are found here. This has a double membrane with pores, and then your nucleolus is this dead center of your nucleus there. That's where the ribosomes are made, okay? So any of this like bold and underlined stuff, protein synthesis, membrane synthesis, perform protein synthesis, produces ribosomes, those are like the key ideas behind these that you're gonna wanna memorize, right? And I think this is our or is this group 4? Group 4, what are we dealing with? Golgi Apparatus. So it forms vesicles to modify, store, sort, and ship. It's like the post office of your cells, right? Yeah, that's the big thing there. And then what's this last one? Group 5 that everyone left? Plasma membrane, is that what you said? It's lysosomes. Lysosomes are for digestion, right? So they take this food, they put it in a vacuole, they digest it, okay? We're going to talk about this in more detail in my super session on Wednesday, so if you're not getting it, don't panic. Also, where'd the roll end up? Did anybody have the roll? Nobody has the roll? Can somebody please bring that roll to me?